What does a pathologist do with your lymphoma lymph node biopsy? When your doctor suspects lymphoma, like non-Hodgkin lymphoma, they might recommend a lymph node biopsy. This small tissue sample holds the key to understanding what's going on inside your body. Once the biopsy is taken, a pathologist steps in to analyze it thoroughly. Their job is vital because they help determine if the lymph node is affected by lymphoma and what type it might be. First, the sample is collected either through a needle, which is less invasive, or by surgically removing the entire lymph node. After collection, the tissue is carefully examined visually. The pathologist notes the size, color, and texture of the tissue, looking for any obvious signs of abnormality. This initial look can give clues about whether the tissue appears healthy or suspicious. Next, the tissue is preserved in a special solution called formalin. It is then embedded in a wax block and sliced into very thin sections. These slices are stained with dyes that highlight different cell features. Under a microscope, the pathologist can see the arrangement and appearance of the cells inside the lymph node. This step, called histology, is essential for identifying abnormal cell growth typical of lymphoma. To get more specific information, the pathologist uses immunohistochemistry. This technique involves applying special antibodies that attach to proteins on the cell surface. By identifying these proteins, the pathologist can determine the exact type of lymphoma. Different lymphomas have unique protein markers, so this step helps classify the disease accurately. Flow cytometry is another tool used in diagnosis. It analyzes the physical and chemical properties of cells in the tissue, such as size and surface markers. This method is especially helpful for identifying abnormal cell populations that are characteristic of lymphoma. Sometimes, the pathologist performs molecular studies like polymerase chain reaction or fluorescence in situ hybridization. These tests detect genetic changes or abnormalities in the cell's DNA or chromosomes, which can be associated with specific lymphoma types. These genetic insights can help refine the diagnosis and guide treatment options. Finally, after gathering all this information, the pathologist combines the results to make a final diagnosis. This diagnosis is crucial because it influences the treatment plan and prognosis. The entire process from biopsy to diagnosis usually takes a few days to a week, depending on the complexity of the tests. In summary, a pathologist plays a central role in analyzing your lymph node biopsy. They examine the tissue visually, stain it for microscopic evaluation, identify specific proteins, analyze cell characteristics, and look for genetic abnormalities. All these steps work together to provide an accurate diagnosis of lymphoma, helping your healthcare team decide the best course of action. Remember, always discuss your results and next steps with your doctor, as they can interpret the findings in the context of your overall health.